These lecture notes are from Professor Deepali Bhandari from Chemistry 441B. So for my mini lecture, I will be going over part 3 of chapter 16, Lipid Metabolism, specifically cholesterol synthesis and metabolism. And by the end of this lecture, we should be able to know how cholesterol is synthesized, know the rate-limiting step in cholesterol synthesis, the enzyme's name, and how it is regulated. We should also know how cholesterol is transported in the body, how cholesterol synthesis is regulated, and know how receptor-mediated endocytosis of LDL, how it works, how defects in this process lead to familial hypercholesterolemia, or FH, and know how statins work to lower blood cholesterol levels. So what is cholesterol? Cholesterol is the most publicized lipid and is notorious for the correlation between its high levels in the blood and incidence of cardiovascular diseases. Although it is often talked about in a negative light, it does have some crucial roles, like being a component of cellular membranes and a precursor for steroid hormones and bile salts. So here we have the structure of cholesterol, which is 27 total carbons, and A, B, C, and D in the structure are 4 carbon rings that are fused, and A, B, and C all have 6 carbons, while D only has 5. It is planar and rigid and a major sterol in animal tissues. It is also amphipathic, meaning that it has a polar head group and a nonpolar hydrocarbon chain. So here we have some facts about cholesterol synthesis. One being humans can synthesize about 1 gram of cholesterol per day and absorb about 0.3 grams per day from their diet. And because of the de novo cholesterol biosynthetic pathway, cholesterol is not an essential lipid in the human diet. And cholesterol biosynthesis occurs in all cells, but the largest amount is made in the liver. So cholesterol is synthesized in 4 stages. The first stage being where 3 acetyl-CoA molecules condense to form mevalonate, as you can see here. And the second is where mevalonate is phosphorylated and decarboxylated to form the 5C isoprenoid intermediate, known as isopentenyl pyrophosphate, which is an intermediate in several other biosynthetic reactions, including chlorophylls and plastoquinone in plants, vitamins A, E, and K quinones in animals. So stage 3 occurs in 2 steps. Part A, where 3 molecules of isopentenyl pyrophosphate are combined to make farnesyl pyrophosphate, as you can see here. And part B, where 2 molecules of farnesyl pyrophosphate are used to generate squalene. Farnesyl pyrophosphate is also used as a lipid anchor in some membrane-associated proteins, for example, heterotrimeric G proteins. In the last stage, squalene cyclizes to form cholesterol, which is a 4-ringed sterol molecule, and this occurs in many steps and over 20 reactions. Plants also synthesize 4-ringed sterol molecules from squalene, but using a different set of reactions. In stage 1, the formation of mevalonate from acetyl-CoA, the rate-limiting enzyme in the cholesterol biosynthetic pathway is HMG-CoA reductase, as you can see here, and HMG-CoA reductase is the target of statin drugs, which are used to treat cardiovascular disease. Cholesterol biosynthesis is regulated at several levels. It is a complex and energy expensive and we regulate the biosynthesis to complement dietary intake of cholesterol. In mammals, cholesterol production is regulated by intracellular cholesterol concentration and by the hormones glucagon and insulin. The rate-limiting step in cholesterol synthesis is the conversion of HMG-CoA to mevalonate by the enzyme HMG-CoA reductase. So for hormonal control, glucagon inhibits cholesterol synthesis and insulin promotes cholesterol synthesis. So there are 3 fates of cholesterol synthesized in the liver. The first being used in cell membranes, the second being converted into bile acids, or the third being converted to cholesterol esters and either stored in intracellular lipid droplets or packaged into lipoprotein particles and exported into the circulatory system. So what is the sterol regulatory element binding protein, also known as SREBP? SREBP is a large protein embedded in the ER membrane as an inactive precursor. And cholesterol binding to SCAP and 25-hydroxycholesterol binding to INSIG results in the formation of a SREBP SCAP INSIG complex that maintains the inactive precursor SREBP in the ER. As you can see here, this is the complex, and in the nucleus, SREBP binds to the sterol regulatory element, also known as SRE, DNA sequences located in the transcriptional control region of specific genes, and then activates the gene transcription of enzymes involved in cholesterol biosynthesis like HMG-CoA synthase and HMG-CoA reductase, and that of LDL receptor to increase uptake of cholesterol from LDL particles. So atherosclerosis is the accumulation of cholesterol in blood vessels, and it occurs when the uptake and or synthesis of cholesterol exceeds the amount needed for membranes, bile salts, and steroids. And as you can see here in the image is a normal artery and a artery narrowed by plaque. So for clinical treatments, statins have been shown to be effective for treating atherosclerosis by lowering serum LDL levels, and statins can also be prescribed to heterozygous FH patients as well as other individuals with high cholesterol levels. Thank you.